Hello everyone, my name is Siti Hajar Zabri and I will be presenting my research titled Characterization of the Reward Structural Connectivity in Female Malay Adolescents using Diffusion Magnetic Resonance Imaging. And my supervisor is Dr. Asma Hayati Ahmad and my co-supervisor is Associate Professor Dr. Rahimah Zakaria, and I am a master student from Department of Neurosciences, School of Medical Sciences, Universiti Sains, Malaysia. So the reward network is the regions involved in processing and responding to reward stimuli. The reward structure connectivity has previously been studied, especially concerning reward sensitivity and addiction. So there were many previous studies. So this is an example of a study that I show you by Vanderbosch and colleagues in 2015. So basically they did a diffusion MRI study and chose the C and targets based on the previous studies which were consistent with the frontostriatal surface, including primates and other human diffusion MRI studies. So they chose the striatum as C and 10 a priori target mass, which are anterior cingulate cortex, posterior cingulate cortex, dorsal ACC, middle orbitofrontal cortex, MOFC, inferior frontal gyrus, supplementary motor area, dorsolateral prefrontal cortex, DLPFC, ventrolateral prefrontal cortex, VLPFC, and hippocampus and amygdala. So the adolescents are of the age of 10 to 25 years old based on previous brain studies. They are known to be associated with higher risk-taking behavior and impulsivity. Adolescence is also often the time of onset for substance abuse. This may be due to the constant development of brain regions during the adolescence period, and adolescents have also been shown to have higher reward sensitivity compared to other age groups. The diffusion MRI is an excellent tool for investigating the white matter integrity connecting the reward regions of the adolescent's brain. So the research questions are, what is the characteristic of the reward structural connectivity in terms of connection strength, which is obtained from the number of streamline in Malaysian Malay adolescents, and what is the characteristic of the reward structural connectivity in terms of parcellation of NACC in Malaysian Malay adolescents. So the research hypotheses are the relative connection probability and parcellation of the NACC to the 6 reward-related regions would show highest connection probability and strongest connectivity to the MOFC compared to the other 5 target regions, amygdala, ACC, hippocampus, VLPFC, and DLPFC. The parcellation of NACC would show more widely distributed pattern of connectivity to the MOFC compared to the other 5 target regions. So specifically, the study objectives are to determine the connection strength from relative connection probability of NACC, which is the seed shown here in the figure, and to amygdala, ACC, MOFC, the hippocampus, VLPFC, and DLPFC. So these are the target regions. To parcellate the NACC according to its connectivity with amygdala, ACC, MOFC, hippocampus, VLPFC, and DLPFC2, and to describe the pattern of distribution. So basically, we got the ethical approval from Japan USM, and the research design is of a cross-sectional study design. We got 15 female Malaysian Malay participants, which are adolescents, and the data was obtained from USM Health Campus, Kubang Kriang, Kelantan. And to ensure the participants had typical reward responsiveness, the reward responsiveness scale, RRS, and the behavioral inhibition system and behavioral activation system scale was given to the participants to answer via online questionnaire. So the DTI database of 15 female Malay adolescents were obtained first. You can see within the study flowchart. And we did the pre-processing of DTI data, contact the participants for online questionnaire via the Google form, probabilistic tractography to obtain the connection probability, calculate the relative connection probability, parcellation analysis via the fossilized FSLIs, and statistical analysis via SPSS. Here I show you an example of graphical user interface of the FSL. Here's a more example of the interface and fossilized application that I use within the current study analysis. Here are the fossilized application. So the results are from the relative connection probability found that the highest relative connection probability of NACC is to the MOFC in both the left and right side of the brain compared to the 5 targets that I mentioned. Result of parcellation analysis showed that the NACC parcellation have the widest distribution to the connection of MOFC. You can see here represented by the beige color compared to the 5 targets in both sides of the brain. This is an example of one of the participants. So the discussion is basically the current studies showed that the highest connectivity of the NACC and MOFC compared to other targets which support the hypothesis, and this finding support that NACC and MOFC have the highest connection strength compared to the 5 targets. NACC and MOFC are 2 key nodes of reward network. The track connecting NACC and MOFC was even distinctively characterized by Casco et al. As a combofrontal track. And this track the finding of this study basically may be attributed to the early maturity of this track. So what they found is that this track matured quite early during mid-adolescence. They found that the average fractional anisotropy of the combofrontal tract undergoes significant changes with age across the lifespan, whereby there is an early peak at age around 14.8, followed by a decrease, then leveled out. There was an even sex difference that they found, whereby this tract of males matures earlier compared to the females, which is around 13.9 versus 18.6. So males mature earlier within this tract. So basically, the conclusion is that the relative probability of connectivity and parcellation showed the strongest connectivity of NACC to the MOFC in Malay adolescents, which are females, and this support the finding of highest connection strength of NACC MOFC of previous studies. Thank you.